let me sort of perpetuate the idea of sharing obsessions with you and share with you my obsession on a disease that threatens the economies of the developed world, and that is dementia. Dementia literally means loss of the ability to think, to mentate. Dementia is now the most feared disease. More than cancer, more than heart disease, more than stroke, people are afraid of becoming demented. And rightly so. 1/2 or more of people over age 85 are demented now or will be demented soon. It's an incredibly common problem. Each person with dementia will cost someone $1 million. We typically think of dementia as Alzheimer's disease, and that's what you see here pictured. What we don't usually think of is someone like Patrick Grange, who died in his late 20s with a brain degeneration known as CTE. CTE stands for chronic traumatic encephalopathy. CTE was first described in the 1920s, but for most of the next century was thought to be primarily a disease, or exclusively a disease, of boxers. And it was known instead under the name of dementia pugilistica. That all changed about 9 years ago when neurologists and pathologists at the University of Pittsburgh studied the brain of Mike Webster, a Pittsburgh Steelers and Hall of Famer who was a brilliant footballer and at the time of his death was living in his car, depressed, psychotic, withdrawn due to CTE. What you see on the right side of the slide there is a nerve cell from the brain of Mike Webster. Our cells operate because the molecules inside float around and talk to each other. In these brain degenerations, rather than floating around, those molecules clump together. So this dark purple stripe, this dark purple process or fiber that you see on this slide represents molecules in that nerve cell that should be light pink and floating around like what you see around it, but instead have clumped together. This is called aggregation. Clumping works just fine, but that's really the basis of all of the late life degenerations that we know about, and also of CTE. Now, if the figures of Alzheimer's disease weren't frightening enough, just last month, the NFL released its formal estimate that about 1/3 of US NFL players will dement, will lose their brains, due to repetitive head injury of sport. This is a striking number, probably larger than even I would have expected, but the truth is we don't yet have the data to understand exactly whether that number is correct or to predict who's at the highest risk. So what we want to do now, and in the partnership between the GE and the NFL, called the Head Health Initiative, is to describe this disease from scratch. Disease hasn't been properly described. And this involves a collaboration between academics, neurologists, psychiatrists, neuropathologists, and the technology team at GE. You see the PET MR at the back of the room is a key feature of what we require to do this sort of research. It takes decades. We need to identify first ways to determine accurately the prevalence of the disease so that we can tell people how risky it is for them to play. We currently can't do that. We don't have the data to do that. But more importantly, if we could predict who's at the highest risk, we might either intervene or at least advise them that this is not a good career for them to contemplate. So in addition to the clinical characterization, and this requires following subjects, patients, over a decade, maybe more, to look for ways to correlate or predict what's going to happen clinically based on what you see in advance in imaging or in genetics. One of the first modalities that have been used is MRI. And you see the structural changes there there are characteristic structural changes, including injury to the vasculature that are characteristic of CTE. The nerve cells in the brain have lots of these long fibers so that one side of the brain can connect to the other and talk to the other. Those fibers are very prone to break during jostling of head injury. So the mapping of these fibers is something that we've been able to do now with MR, but have not been able to do even with pathology in the past. That's the connectivity change that you see depicted there. Metabolic changes, we can now see not only the utilization of sugar, but the buildup of these clumped proteins. So we now exploit the pathology of these diseases to find ways of detecting the pathology either in advance, as is the case with Alzheimer's disease, in which we can detect these changes 20 years before the first symptom. Or in CTE, at this point, we don't yet know whether we can predict CTE, but we can at least diagnose it with these brain scans. This really exploits the differences in the pathology. Alzheimer's patients develop clumps of a protein called amyloid. What you see in the center of the slide here is an amyloid plaque. They're spherical, but when you look at them under the microscope, they're sort of circular. This fuchsia central area is the amyloid material. What you see arrayed on the outside are the tangles. And that's exactly the same structure that you saw before in Mike Webster's brain, these dark purple sort of radiating fibers. Alzheimer's patients develop both amyloid plaques and tangles made of another protein called tau. Usually, people with CTE develop only tau tangles and little or no amyloid. So as we recently reported, a retired NFL player who came to see us because he thought he had Alzheimer's disease, and his wife thought he had Alzheimer's disease. We were able to employ this new technology to find that he had no amyloid in his brain. Most of the clinical trials that we're doing now are with compounds that reduce amyloid. So he would not have benefited from that trial, and the trial would have been contaminated by having someone who didn't actually have Alzheimer's disease. What you see here are examples of these scans, and though they look similar, they're produced by very different chemicals. These are PET scans in which chemicals that have a very high affinity for these plaques or the tangles differentially are infused, and the PET scan is taken of the brain about an hour later. On the right, you see the distribution of amyloid plaques in the brain of someone with Alzheimer's disease. On the left, you see what are now experimental scans using a compound called THK5117, which selectively visualizes tau tangles. So though you get the same sort of pseudo-color image, the ligands are very different. In CTE, the scan on the right would be negative, would be blue. So we can now distinguish the diseases at the clinical level. Together with GE, we're in defining Alzheimer's disease and CTE, not just at the clinical level and at the sort of structural imaging level, but also at the micro-imaging level. Why is that? Well, we know that there's at least one way that these diseases differ in terms of their structural pathology, but there might be others. This slide shows what's called a multi-omics image produced by GE and Niskayuna. The purple or fuchsia spots that you see there are again amyloid, but all these the other blues and yellows in the surround are all abnormal structures that are present to one degree or another in these diseases. And if we can understand why they're there and how they're there, we can develop new ligands, new dyes that will enable us to see these changes, hopefully early on. One mystery has been why it is in CTE there are only tangles and no amyloid plaques. It appears that one sort of the leading hypothesis for why that's true is that every little hit might activate the inflammatory response, that is, brain inflammation. And so GE is now developing ligands, PET chemicals, that will help visualize brain inflammation. So the panels that you see here show these sort of the currently available best brain inflammation ligand, which is the A on the left, and the new GE 180 on the right. So what's called the coronal and sagittal sections are cross sections, and below are 3D reconstructions. So as you can see, these are the same brains. If you look at the brain with the current image, you can barely see any signal at all. However, with the new GE 180, there's a robust signal. We're now beginning to infuse this ligand into subjects that we think have CTE. I mentioned the MRI and the fiber tract tracing, and this shows probably a little easier image to see. And these fibers are largely intact in the image that you see here, but in CTE they become disrupted, and you can actually visualize that during life in ways that we can't even visualize at postmortem exam. So what we want to develop now are ways to personalize the risk, to predict exactly which individuals are at the highest risk for developing these diseases. Why is that? Well, we already have lessons from what we've been doing with Alzheimer's disease, that if we intervene early, before there are symptoms, we might be able to have some impact. That is probably also true of CTE, and we already have a whole sort of panoply of chemicals, of new compounds that have been developed for Alzheimer's disease that may be relevant to CTE. But this has a profound cultural implication. One of the questions I'm asked most often is, should I let my child play high school football? These athletes, this is not just a disease of professional athletes. The youngest person to be observed to have CTE in his brain committed suicide at age 18. So this is a disease that can be acquired during high school athletics. So it's very important that we get on to describing this disease and finding ways to intervene. Thank you very much.